Welcome to Structure Club. I'm Scott Stagg. I'm a professor at Florida State University, and I use cryo-EM to determine the molecular mechanisms of membrane remodeling.
And I am Ashwant Francis, an assistant professor at Florida State University. My lab focuses on the structural biology of viruses and how they interact with host cells. Together, Scott and we have come up with the Structural Club as a journal club podcast and YouTube channel where papers are given by the authors themselves.
Yeah, so each episode, our idea is to bring on a guest to share one of their own papers, and then the episode will end with an interview where our guests can share their insights, talk about their scientific journey, some of the science they're most excited about. Today's speaker is Andres Leschziner. He's from the Weill Cornell Medicine in New York.
Welcome, Andres.
Hi, thanks for having me. Well, good to see you, Scott. We've known each other since we were baby scientists, and nice to meet you, Ash.
Great. Yeah. So on a little introduction, Andres got his PhD at Yale, followed by a postdoc at Berkeley. He started his own lab at Harvard, and then he moved to UC San Diego. And he's currently a professor of biochemistry and biophysics at Weill Cornell Medicine. And he's going to be talking with us about his paper, Structure of LRRK1 and Mechanisms of Autoinhibition and Activation. And that was published in October 2023 in Nature Structural and Molecular Biology. Andres, take it away. Share your screen and take it away.
Let's go. Yep. Okay. You have to allow me to.
Oh yeah, let's do that. Allow. There. Okay.
Cool. I have to do it. Sure. Okay. How does that look?
All right, I see your computer.
Yeah.
And there are the slides.
Okay. Good. Okay. Well, thanks, Scott and Ash. I'll get going right away then with a little bit of background and sort of origin story. So we've been sort of working on the molecular basis of Parkinson's disease for a few years, and I think most of you will know about Parkinson's, but maybe some of you don't know that about 10% of the cases have a direct genetic component, and mutations in a protein called leucine-rich repeat kinase 2, as a mouthful, LRRK2, are the most common cause of the genetic form of Parkinson's disease. So here's a brief sort of history of this protein. So the link between LRRK2, or the gene part 8, and Parkinson's was made over 20 years ago in a Japanese family. Soon after that, people show the mutations in LRRK2 are what causes PD in this type of PD. And soon after that, those mutations increase the activity of the kinase, right? The K in LRRK2. That got pharma a little bit excited because you could treat at least these patients with inhibitors. And then in 2018, another group showed in patients with idiopathic PD, so no mutations anywhere, that their LRRK2 kinase was also hyperactive. So that got pharma very excited because maybe you can now treat a lot of Parkinson's patients with kinase inhibitor. There are actually 4 clinical trials going on right now targeting LRRK2 and more than 30 companies working on these approaches. So we were working on LRRK2 at the time. It's a pretty unhappy protein, very difficult biochemically and structurally. So we wondered whether we could learn about LRRK2 by studying LRRK1. So in a way that, I think this is also a lesson on how you never know where science is going, and the surprises are often the most interesting thing. We thought LRRK1 may be a proxy for LRRK2, right? Maybe it was easier to work with them because, they seem to be very similar. We're going to learn about LRRK2. So let me tell you about sort of compare and contrast. If you look at domain organization, very similar. The main difference is that LRRK2 has this armadillo repeat domain at the N terminus that LRRK1 doesn't have. Their C-terminal half, which is where the catalytic parts are, very similar. They both have a RAS-like GTPase in green and a kinase in orange. So very similar. Their disease association, very different though. So LRRK2 linked to Parkinson's disease, as I told you. LRRK1 is involved in rare bone diseases like osteopetrosis. And their causes are also very different. So in LRRK2, all the PD link mutations are autosomal dominant gain of function, so increased kinase activity. What in LRRK1 is the opposite, the autosomal recessive loss of function. As far as I know, there are no reported gain of function mutations in LRRK1. They both phosphorylate RAP GTPases. This is also RAS family GTPases that live on vesicles in the cell and, broadly speaking, sort of control trafficking. And as far as we know, they phosphorylate non-overlapping sets of Rabs. Both of them use autophosphorylation, and LAR2 had been shown to bind to microtubules, at least under certain conditions, and we showed that LARC1 did not. So this is where things were when we started. I'm going to give you a brief introduction to kinase anatomy because it's going to be important for a couple of the things we'll discuss, and I'll do it in cartoon form. So kinases are bilobe structures. They have a smaller N lobe, the N terminal, and a larger C lobe. An important player today is going to be this alpha helix called alpha C. Now, helix have several important residues. We're not going to be talking about the G loop or the Sol bridge, but we will be talking about this catalytic triad called the DYG or DFG motif. So most kinases have DFG, but LARC1 and LARC2 are in the minority that has a tyrosine instead of phenylalanine. So this is a kinase in the active conformation. And what that means is that the tyrosine is buried in a hydrophobic back pocket there, and that positions this catalytic aspartate for phosphoryl transfer. When the kinase, and what happens then is that this tyrosine stacks on top of all these other residues to form this hydrophobic spine, and this is what stabilizes the active conformation, and residues in the R spine are involved in that. When this doesn't happen, then the tyrosine goes somewhere else, the triad is not in the right place, and actually the green bit is completely disordered, and that's where ATP sits. Okay, so we started by solving the cryo structure of monomeric LARC1, and just as a reminder, they look very similar at the 1D level. All you're missing is the armadillo repeats in LARC2. So this is the structure of the monomer. And I should point out that even though we had the anchoring domain in our construct, it's floppy, and we don't see it in the M-density, that's why it's dim here. But you see that we start here in light blue. The LRR, that's the leucine-rich repeat. That's what gives rise to the name of the proteins. So you go around here, you get to the green, that is the GTPase, then two architectural domains in yellow, and we'll talk about one of them later. And then you go into the kinase in orange, and finally in red, the WD40 domain, the donut shown here in the cartoon. We can skip the models.
Andres, just
yeah.
so to orient myself here. Okay, so the kinase part is phosphorylating the Rabs.
Yes.
And then the rock part, it's involved in regulation of this complex. Is that right?
Yes. So yes, we're actually just about to submit the paper explaining how this regulates LARC2, which we think is going to be the same in LARC1. We can talk about that later, but yes.
Okay, cool.
That had been a mystery for a long time. Why do you have a GTPase there?
Yeah. Okay. Awesome.
At this time, we had no idea.
Okay.
Yeah. Okay. So let's compare now LARC1 and LARC2 at the monomer level. Again, this is the model that we built of LARC1. And while we were working on this, the structure of full-length LARC2 came out. So we had solved the first structure of the C-terminal half of LARC2. And actually, we had the structure full-length, but we got scooped by another group while we were writing the paper. But we knew already about the structure. And we knew that the leucine-rich repeats, again, in light blue in LARC2, they go right above the kinase active site. So they physically block access to it. So, small molecules can get there, no problem, but a Rab could not make it to the active site. So this is the autoinhibited state of LARC2, which we now understand well. But LARC1 is not doing that, right? The repeats, they're there, so obviously they must be doing something, but they're going down and away. The kinase active site is completely open. So we were like puzzled, right? Because why do you have that there if it's not doing the one interesting thing that LARC2 seemed to be doing? And this is just emphasizes that again. So LARC1 in light blue, LARC2 in dark blue, very different positions for an element that is conserved in two. We're going to take now a look at three different sort of structural elements in LARC1. So the first one is this one shown here as B, and we're going to be looking at the long helix here in the kinase, which is that alpha C helix I mentioned earlier. I remind you again in the cartoon. So it sits here and it's involved in forming the spine and does other sort of interesting things. So it's super long in LARC1. So this is LARC2 by comparison, and this is a superposition. So LARC2 in dark, LARC1 in lighter colors is at least twice as long. And when we looked at the time, I think this was the longest alpha C helix ever seen in any kinase structure. I wish I could tell you why is that long. We have no idea, but it's clearly unique. The second thing we're going to look at is the WD-40 domain. Remember the donut in the back? The strands don't matter here. What's interesting is that LARC1 has this giant loop, 120 residues, completely disordered in our structure. We didn't see any of that. And that is not present in LARC2. LARC2 is different in that region because the repeats, the long arm comes around, and this helix here called the hinge helix, at the top you can see it here, is basically involved in stabilizing this long arm, attaching it to the WD-40, and that's how you keep the uninhibited conformation sort of stable. And it doesn't have this giant loop. So again, why such a big loop? We'll see later. Again, comparison there, it doesn't matter. And now we're going to look at the kinase active site from the back, so from behind the screen. And so again, this is the kinase, this is the active site. And here we have the darker yellow domain called core B, C-terminal of ROC, and ROC is the RAS of complex. Sorry about the names. But so this core B also has a long loop, about 40 residues, completely disordered in our structure, and that is not conserved in LARC2. LARC2 has 5 residues that are not seen in the structure, so again, no idea here what this loop might be doing in comparison. But the main point to take from here, big loop here that we don't see, big loop here that we don't see. So then we solve the dimeric structure of LARC1. That's when some of these mysteries become to be solved. So this is the dimer. We call it the yin-yang dimer, hopefully for obvious reasons. So you can see that this is one monomer. You have the leucine-rich repeat and the anchoring domain here, wrapping around and going toward the other one. And this answers the first big question. LARC1 also has autoinhibition, but it does it in trans. So instead of blocking its own kinase, it goes around and it uses the anchoring domain to block the other monomer's kinase. So it does what LARC2 does as a monomer, but it does it as a dimer. So that solved that question.
So is it then, do I understand correctly that it's the leucine-rich repeat in LARC2?
So this would go like here in LARC2 and cover
that's like flipping it
kinase.
up.
Yes.
And that's like interfering with the kinase?
So in LARC2, it just blocks it. It basically, you can't get to it. And here what it's doing is the LRR is what reaches over, and then it's because it's too far away, it uses the anchoring repeat to do the same thing. It just blocks it. You can't get to it.
Cool.
Model, again, that doesn't matter. The main point, you can see that from the map. Now we can compare the dimers of LARC1 and LARC2. So this is now a surface representation of the molecular model with Biol for LARC1 and the dimer for LARC2. And hopefully it's obvious that they're very different. LARC1 is an antiparallel dimer with a twofold axis perpendicular to the screen. LARC2 is a parallel dimer with a twofold axis parallel to the screen. And now we're going to look at all the interfaces that stabilize these dimers. We're going to start with an anchoring interaction in LARC1. And the dimer had a slightly lower resolution than the monomer. So we build the molecular model in the monomer. And in the dimer, we can build side chains everywhere. So here we see a continuous density between the two anchoring domains. There's another interaction between the anchoring of one monomer and the leucine-rich repeat of the other. And here you see a nice charge complementarity. This guy, the anchoring sort of nudges against this cavity that is negatively charged. There's a tripartite interaction here. So leucine-rich repeat and kinase from one and anchoring from the other. Again, a lot of continuous density there. And then one that actually bridges the two kinases, which are at the core of the dimer. So again, we see this nice bridging density between the two kinases. And the final one is one between the kinase of one monomer and the anchoring of the other one. Again, continuous density. Now, LARC2 has a single dimer interface that it involves the darker yellow domains, the core B domains. The anchoring repeats here. Even though they seem like they touch, if you rotate this by 90 degrees, there's a big gap between them. So only one interface as opposed to already 5 interfaces I showed you here. And here's now the map again, then rotated 45, another 45, and then I cut through it to the middle, and you can see the sandwich here where the anchoring repeat sits right above the kinase active site. And so the kinase is really buried in the middle of the dimer and covered by the anchoring repeat. There's no way a protein can get to that. Okay, so let's now start talking about all the disorder loops I've mentioned and one I didn't mention. So this is again the model of the dimer from like a side view. This is the anchor and repeat. And we started building at about residue of 50. This is an AlphaFold 1 model, so hopefully the models will look better now. This is a while back. But the first 48 residues seem disordered, and they're not in our model. Our first residue is arginine 51. But when you look at the map, and again, what's colored in magenta is disordered, is stuff that was not accounted for by our model. And it has about the size that you would expect to accommodate a lot of the missing residues. So we're wondering whether this is an interaction that is involving stabilizing the dimer and so promoting autoinhibition. So if that's true and you remove this interaction, maybe you can activate LRRK1. And that turned out to be true. So the controls here are this mutant is a kinase dead, so it should have no activity, and it doesn't. This mutant basically is the equivalent of a Parkinson's mutation in LRRK2, and it's activating. And then this is wild type. And here's, even if we delete the first 25 residues of the missing part, we get an activation that is equivalent to the Parkinson's-like mutant. So this is consistent with the idea that this is stabilizing the dimer to promote autoinhibition. Now, if you also again look at the whole map and you lower the density, we see this like kind of mushroom-like thing connecting the two WD-40 repeats. This is low resolution, super noisy. There's no hope of building anything there. But remember that I told you that it's a very long loop in the WD-40. So 120 residues, about 110 are modeled here again by AlphaFold that are not part of our model because they're outside the density. So maybe this is equivalent. Maybe these loops are coming together to make the mushroom and also stabilize the dimer. So same experiment, we deleted those, and we see the same thing. Even though the activation is not as high as what we see with the deletion of the N-terminus, we still have a statistically significant increase in activity. Oh, and I should have mentioned, what we're measuring here is phosphorylation of RAP7A. This is one of the physiological substrates of LRRK1 in cells. So these are experiments in cells. Okay, so I'll talk about what I think is the most interesting loop, and that's part of the dark yellow core B domain. So now we're looking at the map of the monomer in the context of the dimer. Remember that this loop was the loop I told you was completely disordered in the monomer. There were about 40 residues missing. In the dimer, we see it. And you can see now in yellow, this density sneaking right into the active site of the kinase. We're going to zoom in now into the dash square there. And now I'm showing you the model. Again, orange, the kinase, in yellow, the core B. And you can see that at the tip of the loop, we have this phenylalanine. And what the phenylalanine is doing is now bearing into the hydrophobic back pocket of the kinase. Remember that I told you here that for a kinase to be active, the tyrosine or phenylalanine of the DYG or DFG motif has to dock into the hydrophobic back pocket for the spine to form and the active conformation close of the kinase to be stabilized. So here, what LRRK1 is doing is plugging its own hole with this loop and preventing the tyrosine here, tyrosine 1410, from going to the back loop. So now this is stuck in an inactive conformation. So if that is true, if you remove the plug, you should be able to also activate LRRK1. And that was true. A single mutation turning phenylalanine into alanine now gives us a twofold activation, again, equivalent to the Parkinson's-like mutation. Now, if you look to the loop in sort of at a bigger picture, there are 3 residues here, 2 serines and a threonine, that have been shown by another lab to be sites of phosphorylation by another kinase. And that lab had also shown that if you mutate them to alanine, you inhibit LRRK1 activity in cells. So our hypothesis was that when you phosphorylate these residues in the cell, you structurally blow up the loop, you remove the plug, and now you allow this tyrosine to go in and the kinase to be activated. So we tested that first by combining all 4 mutations. So here we have the removal of the plug plus 3 phosphoromimetic mutations. So we turn all these 3 into glutamates. Now we have more than a 3-fold activation of LRRK1. And then we combine only the phosphomimetic. So we kept the phenylalanine that could plug if it made it to the hole, and combine that with the deletions of the N-terminus. Remember, the first few residues of the anchoring, the repeat that we showed, are important for the dimer. And you have an increase that is small, but is statistically significant. And this tells you that there are two levels of autoinhibition in LRC1. You have the dimer, so you have to first to break the dimer to get to the monomer, and then the monomer has its own autoinhibition that has to be released as well. So remember that it's all started when, LRC1 was going to be a proxy for LRC2. It was clear by now that it was not a proxy. It was very different in interesting ways, but we were learning about LRC1, not LRC2. But this is sort of a win-win situation, because now what we could do is, by having structures of related but different proteins, we could do an evolutionary analysis of the structural motifs in LRC proteins in general. So we teamed up with Matt Doherty at UCSD. He's a molecular evolutionary biologist, to do an analysis of all LRC proteins. So there are 4 LRCs in nature, LRC1 through 4. We vertebrates have only LRC1 and LRC2. Nematodes and arthropods have only LRC3, and this is a point that we raise in the paper, because there are a few studies using either Drosophila or C. elegans as models to understand Parkinson's, and the point we made is that clearly the LRC proteins are not proxies for each other, and so if you want to understand LRC2, you need to study LRC2, and if you want to understand LRC1, you need to study LRC1. So these are the structural elements that we focus on. The first one is that really long alpha C helix, remember, where LRC1 seems to be unique. The second one is this, the autoinhibitor loop with the plug and the phosphorylation sites. The third one is the giant loop in the WD-40 that forms that mushroom density. And finally, and I didn't talk about that, but I mentioned that LRC2 binds microtubules. We saw the structure of LRC2 bound to microtubules and identify what we call basic patches. These are short stretches, very rich in basic residues that are necessary for this interaction. So we wanted to look at those as well. So these are cartoons of the stretches of the proteins we looked at, and the sort of pastel rounded rectangles are where the sequences that where we did the analysis are. And by we, I mean, Matt and his people. Okay, so we start with the WD-40 loop. This is clearly a LRC1 specific feature. It's very huge in all LRC1s and much shorter in all other LRCs, 3, 4, and 2. The Corby loop that we talked about also, not only is it LRC1 specific, is it specific to vertebrate LRC1s. And the 4 squares here are the elements we discussed. F is the phenylalanine of the plug, and P's are the 3 phosphorylation sites that are involved in removing the plug. And the basic patches that I told you about here, the 3 patches that we identify are only present in vertebrate LRC2s. There are 2 in lower LRC2s and only 1 in LRC3, but we don't know whether LRC3 binds microtubules, so we don't know whether this is functionally related to the ones here. And finally, the crazy long alpha C helix seems to be present in LRC1 as well as in LRC3, but clearly much shorter in LRC2 and LRC4. So what's nice about this is that now it gives you sort of a parts list of what structural motifs you should focus on in terms of their potential for being involved in disease. So something unique to LRC2 is more likely to be involved in Parkinson's than something that is not conserved and is present only in LRC1 or LRC3. So summarizing and sort of then circling back to the beginning, what I showed you is that LRC1, the active site is autoinhibited, but it has a different autoinhibition that uses that loop that goes into the active site, while LRC2 uses a direct steric autoinhibition in the monomer, which just covers its own kinase active site. But now LRC1 has this kind of baroque dimer where now one monomer sterically inhibits the other one in trans, and LRC2 has a relatively simple dimer interface, and that's not, we think it plays other functional roles, but not in terms of what I told you about today. Again, simple dimer, one interface. I told you about at least 7 interfaces that we characterize for LRC1, so really a huge effort placed in keeping this dimer a dimer. And we think that this, this is what we speculated in the paper, that this may be related to what I told you at the beginning, right? That there are no gain of function mutants for LRC1. The disease is all loss of function. And I think it may be that it's very deleterious to have elevated LRC1 activity. Obviously, you can have elevated LRC2 activity. It leads to Parkinson's, but it's a very late onset disease. So I think that maybe this is telling us that a lot of effort has gone through evolution to keep LRC1 off, and that's why we see this in terms of the disease. With that, I wanted to thank the people who did the work. There's been a collaboration, with 3 other labs and a lot of fun. Thank you.
Wow, Andres, that was fantastic. I really enjoyed it. I have
Thank you.
a couple of questions
Yeah,
for you
yeah.
regarding. So, evolutionarily, it seems like it was favorable to lose the armadillo domain, right? Of
Uh,
LARC2 to become.
yeah, so that yeah, so I didn't that has to do more with LARC2 and things we don't understand. So the armadillo domain in LARC2 is involving recruitment of LARC2 to memory. So it has as of last count at least 3 binding sites for RABs that are not substrate binding sites, they're recruitment binding sites. That's how LARC2 gets to membranes and sort of is trapped there because it has some sites that favor phosphorylated RABs. So as it phosphorylated RABs, it strengthens its own recruitment. We know nothing about recruitment for LARC1, so we don't know how it gets to membranes. So that must be somewhere else because the armadillo is gone. So whether it's in the anchoring, we don't know. Yeah, but
Yeah.
that's interesting.
So the other thing, is you kept showing your beautiful pictures of the monomers, and
Yeah,
it was so striking how similar these two proteins were.
yeah.
And the armadillo domain, got me curious to think, what if you put the armadillo domain of LARC2 on LARC1?
Yeah, yeah, yeah.
What happens there?
Yes, yeah, totally. Yeah, yeah. That's a good idea. So that's something that we thought. So there are no structures of these proteins bound to RABs. So I wanted to do, I guess it's sort of a similar idea, but if we understood that, I wanted to swap the RAB specificity, and
Yeah,
so see, basically the question is the disease simply at which RAB is phosphorylated, or is there something else about
right.
Yeah, but yeah, no, that's a fun experiment. Yeah,
I think that'll be fun. Yeah, it's
yeah,
it's, it's so
yeah.
exciting. Yeah, thanks.
I have several things that are springing to
Yeah,
mind. One is just like a structural biologist question. Uh, that that looked like it was real pain to model. Like there's, you're
yeah.
like
Also,
how did you how
the dimer that it looks flat
yeah.
loves to have that orientation. I mean,
Uh
we had to do some tilted data, all kinds of crazy stuff. I don't think if it ended up working with this, but we started using structuring affinity grid
really?
really because of that, and they ended up working for other projects. But yeah, it was horrible. Yeah.
Yeah.
Yeah, I think that the more, I mean, the postdoc Janice that worked on that was just amazing. So she could do things that most mortals can't.
Do you think would it have been even possible to get the atomic model without like an AlphaFold sort of to sort of guide your
Oh,
interpretation?
there was no AlphaFold because at the time, yeah, no, we only modeled that little bit. The rest we modeled.
Oh wow.
Well, remember that we had solved the structure of the C-terminal half of LARC2. Right? So we had them all. We could start with that, and then we just needed the repeats. There are a gazillion structures of losing rich repeats, and so that
Sure.
part, yeah. So we're not starting from zero. Yeah,
Got it.
yeah.
And the other thing I was sort of just thinking of wondering about is like someone who's interested in molecules is why do you think it involved this double inhibition? Like, do you think like one of them's just like keeping it from turning over GT or ATP or something like that?
No, I think I think it must be really bad to phosphorylate the RABs that LARC1 phosphorylates. Which is why, because I've always been looking, there must be some gain of function mutant somewhere, and I haven't been able to find one. So I think there must be something about they're in different parts. So I think the LARC1 is more in the endocytic pathway, and LARC2 is more the trans-Golgi. So I think one must be I mean, obviously, Parkinson's is a terrible disease, but you can have hyperactive LARC2 for 50, 60 years before symptoms, right? So that
Mm.
can be tolerated. And there's some data now arising where LARC2 is involving the lung as well. So the someone mentioned recently, there might be some evolutionary pressure where it's good to have a little hyperactivity of LARC2 somewhere else. It's just not good in your brain, but obviously the selective pressure against that is low because it's late.
Oh, that's very interesting. So, the expression levels of these two, LARC1 and 2, in different tissues are different, is what you're.
Yeah, yes.
So
So
both of them, yeah.
So LARC2, and this is always confusing, it's not that highly expressed in dopaminergic neurons, right? Which is where the death of those neurons is what causes the bulk of the Parkinson's symptoms. It's highly expressed in lungs, kidney, macrophages, and microglia. And LARC1 in bone and osteoclasts, which are like the macrophages of the bones. Yeah.
I see. I see. So this type of differential expression is probably that's why it's still maintained, although half of it seemed to have lost that armadillo domain and stuff,
Yeah.
maintained to do different things in different tissues and different cell types.
Yeah, but at the molecular level, they have to be the same. They have to be recruited to membranes, to the wraps, and phosphorylate them. Again, how LARC1 does that?
That's that. Yeah.
Yeah,
Okay.
no idea. Yeah.
So you showed that LARC2 is binding to microtubules, and so presumably it's going along for the ride, like it's being trafficked. No, it's just getting there, and
yeah. So this was shown. So basically, so far it's only been shown in our expression system. So where the concentration is relatively high, and it forms filaments, like beautiful filaments on microtubules. So I was shown initially with a sort of plastic embedded tomography. You could see it was coating microtubules. Then actually Elizabeth Villa showed a sort of cryo-ET in situ structure, where you could see the helix. And then we solved it in vitro, I mean, using helical approaches. That's how nice it is. So, we also showed using this is for LARC2, single molecule methods, that even low levels of LARC2 can stop motors on a microtubule by acting as a barrier. I mean, the jury is still out whether, if you are in, let's say, in a lung cell that expresses it highly, most Parkinson's mutations increase microtubule binding, and certain types of kinase inhibitors also increase that. So if you had all of that, would that be a problem? And that, that question hasn't been answered for technical reasons yet. So we don't know whether the microtubule binding is physiologically relevant, even in a disease state.
I see. Wow. So there's a lot to do over here. This
Yes.
is a great place
No,
to check out.
it's a fun project. Yeah.
It's a fun project. Yeah, that's fantastic.
All right. So that was great. Really cool stuff. Really exciting. So now I think we'd like to switch it up a little bit and sort of interview you to sort of think about your inspirations and stuff. So Ash is going to take it away.
Yeah, we have a few questions for you about this paper, how this paper came together, and more broadly about your journey and how you became interested in this field.
Yeah.
I have the first question, and Scott and I will take turns after that. So one of the things that we want, will be very curious for, all scientists when they see a paper, right? What was the breakthrough moment for the paper? As in, like, when did you know, oh, wow, this is the story. Let's go for it.
For that, it was the dimer, right? Because remember, I told you we got scooped. So Jan is actually the first author of this paper. She had solved the monomer for LARC2. We know it was autoinhibited. That was super interesting. How is that regulated? And LARC1 was sort of the backup plan. And now it's not doing autoinhibition at the monomer level. So now it's like, it's not doing the one interesting thing that full length LARC2 was doing. So and then she saw the dimer and it's like, oh, cool. Okay, it's doing the same thing in a very different way. So now there is a story. So I think that was the moment where it's like, okay, they're similar, at the primary sequence level, but then once you got more complicated, they're very different. And then the autoinhibition came later, and but I think that when she showed me the dimer, I was like, okay, cool.
Wow.
Yeah.
So I think you've already answered this a little bit, but can you sort of place this paper in your scientific trajectory? I know your wife is a co-author, so maybe there's an interesting story there as well.
For me, I think, I'll separate. For me, it's sort of like the midpoint on, my cryo-EM career. I think, Scott and I grew up in the dark ages of cryo-EM. So, the things we can do today, I was hoping we could someday, but I wasn't sure, right? And this is sort of like when, I wanted to cryo-EM because I was interested in conformational dynamics. I wanted to be able to see big things changing shape. And I thought that cryo-EM was going to do it someday, hopefully. We were doing balloon animals at the time. So this is more like what, we're starting to see a couple of things. So I would say sort of the midpoint where we're starting to understand things at this, at the chemical level, which is not true when I was training and things were really low resolution. Now we've moved to where we actually make systems heterogeneous on purpose. So if we're studying an ATPase, we put ATP. We don't try to trap it into any state. The last, the paper we're finishing on LARC2 we solve 16 structures at once on purpose, because we, in this case, we're studying the GTPase. So don't add, only, GTP gamma S, or, throw everything, throw the kitchen sink, and then solve all the structures, and then go look and see what nucleotide is bound. So, and yeah, with my wife, we started collaborating when soon after we started our labs at Harvard. She was at the medical school, and we started working on dining, which is something she did her postdoc on. I was always interested because it's a giant thing with a lot of moving parts. And we said, once we start our labs, we'll start working on that. And basically, we got interested in LARC2 because of the microtubule angle. So I had actually promised myself never to work on a kinase a long time ago. But a colleague of mine came and said, "Oh, I'm working on this kinase." I was like, "Oh, okay." And it's been shown to bind to microtubule. I was like, "Oh, that sounds, it doesn't look like a microtubule binding protein. So what's it doing there?" And that's how we got into that, and then explode. So between her lab and mine, we may have like close to a dozen people working on this.
Wow. Okay.
Yeah.
It's quite
Okay,
a journey,
so-
yeah, from microtubules and dining.
Totally.
how you
Yeah,
got
yeah. But that was the angle, right? We were like, "Oh, the microtubule thing is interesting."
Yeah. So we've been talking a little bit about
Yeah.
retrospective. So I think all of us, once we get to this stage, like there's sort of like a defining moment. Like you've had, like you're collecting data, you're like beating your head against the wall, and then suddenly it appears. Like so, so the question is there a piece of data or an image or something that's sort of like burned into your memory as like the moment that you discovered something important?
Yeah, that was also a structure that someone solved in my lab. We were working. There are two that are sort of tied, but I'll pick one. We were working. My lab had worked on like chromatin remodelers for a while. So this is DNA translocases that massage nucleosomes basically. And we're working on one in collaboration with Dong Wang at UCSD. That is called an orphan remodeler because it's not supposed to really work on nucleosomes. It works on RNA polymerase and helps it bypass lesion. So we were, solving the structure, and no one really knew structurally how it worked. And this postdoc shows me the structure and the RNA polymerase on a transcription bubble with this protein bound to the DNA behind it and bending the DNA by 90 degrees from its normal place. I was like, done. Okay, we'll start writing the paper now.
That's it. Yeah, that's the one. Yeah, you got it.
I was like, I think I said congratulations.
Right.
Your paper. Yeah.
Hey, so who's your scientific hero? Like as you grow up from your school days, college days.
Yeah.
Now,
So I'm gonna cheat and pick two because they occupy sort of the same place in my brain. Then they're from later. I would say they're from grad school, and I would say Richard Henderson and Bob Glaser. For more than obviously the amazing contributions to cryo, but I think again this will resonate with Scott. In the dark ages, again, it wasn't clear where this was going, right? And they were the ultimate cheerleaders, super optimistic, encouraging everyone in the field. And I think at the time, what it sort of missed, we felt we were all on the same boat rowing together, right? Because it wasn't clear how far we were going to get. And they were super encouraging, backing this up with, math and physics, right? I think what got me into cryo was a review that Richard Henderson wrote in 1994. I was
the paper, right?
Yeah, I read it. I was like, yeah, this makes sense. I believe it, right? I was like, only the engineers have to figure something out. Everything else should work out. So that for me, they were very formative, both as examples of collegiality and sort of brilliance. Creating the field, welcoming everyone, and pushing the field.
right.
That's great. All right. Well, Andres, I think we've taken up enough of your time. Thank you so much for joining us today.
Welcome.
So we really appreciate you taking your time to tell us about your science.
This was a lot of fun.
That's great. So thank you.
It was so much. Yeah, it's so much nice meeting you on Zoom and listening.
Likewise.
Science. Yeah, fantastic.
All right. So I think that does it for this episode. And to our audience, we hope you'll join us again for the next episode of Structure Club. Bye.